Hello students, in this practical video we will learn how to retrieve nucleotide sequence from NCBI. As people know, nucleotides are the biopolymers or large biomolecules essential to all known forms of life. The term nucleic acid is the overall name for DNA and RNA. So when we talk about nucleic acid, it may be DNA or it is RNA. They are composed of nucleotides, which are monomers made up of three components: a 5-carbon sugar, a phosphate group, and a nitrogenous base. Although first discovered within the nucleus of eukaryotic cells, nucleic acids are now known to be found in all life forms, including within bacteria, archaea, mitochondria, chloroplasts, and viruses. So, in this practical, we will try to find some of the nucleotide sequence through NCBI. For searching nucleotide sequence, we first have to go to NCBI's homepage. Then we have to search nucleotide database for any organism. In the next slide, I will show you how to select nucleotide database from the NCBI database and how to search a particular nucleotide sequence for a specific organism. So in this slide, I will show you how to select nucleotide database in NCBI's all database. If you see in this slide, in the left hand side, which is marked in red in color, the NCBI homepage is present, which is ncbi.nlm.nih.gov. So when you go to NCBI's homepage, you will see a particular option called as all database. So in all database, all the databases related to protein or nucleotide or other literature database or chemical compound databases are present. So as we need nucleotide in this particular practical, for this we have to go to nucleotide in the list. So near to the all database there is one combo box. So you click on the combo box, one list will come. So from the list you just click on nucleotide, and then in the blank option you just write any organism name, then you click on the search button. So in this slide you can see the result for the search we made in the previous slide. We have searched nucleotide for human, so we are getting lots of nucleotide for human, which may be DNA or RNA. These results, they are having some particular accession number, and they are mainly present as a Jinbang format. So when we see nucleotide sequence in NCBI, we always find them as a Jinbang format, but we can convert them into other format also, like FASTA format also. So out of the different result we have got in the previous slide, I have selected one result, which is Homo sapiens ribosomal protein L34 transcript variant 1 mRNA. This particular mRNA or nucleotide sequence is present as Jinbang format, which is marked as red color in the left hand side. So Jinbang format always start with locus. So if we see Jinbang format, which is for protein, they always start with one word called as locus, followed by some definition line, then accession number, person, keywords, and source. Like this, lots of description line you can see. So if you want to keep them in Jinbang format, you have to have this locus word. If you want to see this particular sequence in simpler format, you can do, or you can convert them into FASTA format. So in this slide, I am showing you FASTA format of the same sequence. If you see properly, you can see that the sequence is very simple sequence. Means in FASTA format, the sequence is very simple. It only starts with a greater than symbol followed by a description line. After the description line, there is no other description. It directly starts. So if you want to save this particular sequence in FASTA format, you have to go to some other option. For saving the sequence in FASTA format, you have to go to send to option. When you go to send to option, you click it, it will give you one more option called as send destination. When you click on choose destination, it will give you one more option called as download file in which format. Okay, so it will be like FASTA only. Then you just click on create file. When you click on create file, automatically it will save the sequence in FASTA format in your desktop. So practice it. Thank you.